So for chapter 4 material, this is when we start looking at cells. We kind of move away from the microbiology to biochemistry. So some people like this because we're moving away, while some people like histology, which is our slides. Some people don't. So this is when our lecture starts to match up with our lab, right? So I wanted you guys to look at the lab manual to look at the type of organs, the type of tissues. Now in chapter 4, we're going to talk about it, and I want you to then use this as a review of the histology. So let's take a look at chapter 4 right here. When we're looking at it, we're going to see that all the tissues in our body are made out of, right, comprise of 4 tissue classification. We have the 4 tissue classifications. There's epithelial tissue, connective tissue, muscle tissue, and nervous tissue. These are the 4 main types of tissue. Now, they all come from embryonic tissue. Obviously, everything comes from embryonic tissue. And remember, we all started out as one cell, one fertilized egg. You can imagine as one cell becomes 2, 2 becomes 4, as they grow, you're going to have a cluster of cells in a ball-shaped mass. You're going to have cells that are part of the ball that's going to be on the outside, right? That's called the ectoderm. The cells on the outside forms the skin. And the weird one is this. It also forms the brain and the spinal cord. So the skin, it kind of makes sense, right? Ecto means outside. It forms the most outer layer of our body, the skin. But the weird thing is also forms the most important as well, internal layer, the brain and the spinal cord. We have the endoderm that's really deep inside of our mass of cells. The endodermal cells forms a lining of the digestive tract and its derivatives, right? Obviously, your internal organs are very deep in our body, and the lining of this digestive tract is even deeper in our body. Now, everything else is from the middle layer of our cluster of cells, right? The middle layer is called the mesoderm, and they'll form all of our connective tissue, including as well muscle tissue, bone tissue, which is connective tissue, and our blood vessels. So mesoderm is very important. It forms a lot of the tissue that helps us, be alive. Now let's talk about the first type of tissue, epithelial tissue. Epithelial tissue consists almost entirely of cells, right? It covers body surface, forms glands. Now with epithelial tissue, it has a free surface where it makes contact with the environment, where you don't have other cells attached to it. So here, this is the free surface over here, right? It has a basal surface. The basal surface is where the cell is anchored to the basement membrane, the foundation. And then it's got a lateral surface where it makes contact with other cells. Now, we just talked about the basement membrane. The basement membrane is the foundational glue that allows the cell to be held in place. Without the basement membrane, any movement on the cell will cause the cell to rupture. There are diseases where the basement membrane and some of the foundational structure and proteins in the epidermis are mutated. So then you'll have babies. Mom will try to pick up the baby. As soon as mom's finger touches the baby and moves the baby's skin, right, to pick the baby up, that little movement can rupture the epidermis in that area, causing painful blisters. So you might have seen pictures of baby being completely covered in what looks like a little gauze. So those babies have that condition where the underlying structure is mutated, and because of it, any movement of the skin causes blistering. So that's important, that basement membrane. Now, other important thing is this, right? It's avascular, right? Your epithelial cells are avascular. Now, as part of the epithelial cells, we're going to see stratified squamous cells are type of epithelial cell. We see stratified squamous cells in your skin, in your epidermis, right? That stratified squamous cells is epithelial cell cluster, and you've gotten maybe a paper cut before. When you got a paper cut, we're going to see stratified squamous cells. They have a lot of layers. That's why we say they're stratified. Right? So let me show you the picture of stratified squamous cells right here. Right? So what happens with stratified squamous cells? There are multiple layers. So these layers have pain receptors. So when you get a paper cut, it hurts. But a paper cut is not deep enough to go where the blood vessels are. So with a paper cut, it might not be deep enough to cause bleeding. In order to have bleeding, you have to go into the capillaries, right? You need that cut into the capillaries. Sometimes a paper cut is superficial enough to cause pain, but not bleeding, right? So being avascular is very important. Now, you do have regeneration up to a certain point. So when we have the epidermis being burnt because you have a sunburn, you can regenerate. But if you have a third-degree burn, not a sunburn, but a third-degree burn in your arm, right? You have oil spill on you, hot oil, that can cause third-degree burns. Well, at that point, you're not going to regenerate. You might need a skin graft. Right. Now, what else are we going to see? In terms of the basement membrane, the basement membrane right here is going to be extracellular, formed by secretions of both the epithelium and the connective tissue. It is an acellular glue foundation, right? It allows our epithelial cells to be connected to the connective tissue. So then it's anchored in place. It can also guide cell migration during tissue repair and healing. And in the kidneys, this basement membrane acts as a filter. So then we can filter out certain things from our bloodstream. Not every epithelium has a basement membrane associated with it, right? Basement membranes are usually thick. So in areas where we want light to go through, we're going to have cells with very little basement membrane. So functions, I don't have anything highlighted because we're going to talk about the functions specifically when we discuss this specific type of cell. So just a few more minutes, we'll go through 110, right? In terms of how we classify epithelium, right? This is pretty easy, kind of remember, right? The big thing is simple, is just one layer. Stratified are multiple layers of cells, with the big one being this, the shape of the cells at the surface or the apical layer is used to name the tissue. So going back here to our stratified squamous cell. If you look at the stratified squamous cell, this is the apical free surface. This is the basal surface where the basement membrane is. If you look at the cells next to the basement membrane, the basal cells, the stratum basal cells right here look like cubes, right? So shouldn't it be called stratified cuboidal cells? No, we call it stratified squamous because we look at the top layer. The top layer of cells look squished, look squamish, right? We name stratified cells based on what it looks like at the very tip, on the very apex. Pseudostratified appears and looks like you're going to have multiple layers, but that's not the case. All cells in pseudostratified epithelium, all cells make contact to the basement membrane. Because they make contact to the basement membrane, they're not truly stratified. Again, once we talk about the stratified squamous in a second, you'll see that there are cells anchored to the basement membrane, but there are a lot of cells that are floating away from it. That's no longer making contact with the basement membrane, right? In pseudostratified, all of the cells there are connected to the basement membrane. Because of it, it's simple, right? So pseudostratified looks stratified, but in reality, it is a simple layering of cells. Only one cell, right? And that's it, one cell layer. All right, let's talk about the specific type of epithelial cell, right? Simple squamous epithelium, right? Here is just one layer. That's where we get simple. And the cells are very thin. Single layer of flat cells, usually containing smooth surface, location are the ones I want you to know are highlighted, right? It lines the blood vessels, lines the lymphatic vessels. It also forms the organ of the lungs, the functional cells of the lungs, called alveoli. So the alveoli are simple squamous cells that inflate. As it inflate with oxygen and atmospheric gas, right? You can imagine that the simple squamous cells, they're already pretty thin. And as you spread it out, it becomes even thinner, right? The thinner the simple squamous cells, the easier it is for gases to move through. Perfect. That's why we want simple squamous cells forming our alveoli, because it's thin, because it allows for ease of gas diffusion. So the function makes sense. Diffusion of gases, diffusion of, right, even inhaled particles, filtration when we see it in the kidneys, right? Again, it looks flat. So here, when we're taking a look at this picture, right, not these big cells over here. That's not the simple squamous. The simple squamous cells are these over here on the sides. You can see the nucleus right here, but you can see the nucleus. And if you look at the edges, look at how thin the plasma membrane is here. It's very thin, right? Because it's so thin, it makes it easier for filtration in terms of kidneys and in terms of blood vessel diffusion of gases. Simple cuboidal cells. Simple cuboidal cells are found in the kidney tubules. They're also found in your endocrine glands. And these simple cuboidal cells right here are going to, right, especially in the endocrine, we find these type of cells in the thyroid, the pancreas, and the liver. So these are places, locations I want you to know for the lecture exam, right? Single layer of cube cells. Some have microvilli, some have cilia. All right, so, all right, I want you to know location and function. Usually for function, it allows us to secrete hormones, right? Like secreting hormones for our, like EPO, that allows us to make more red blood cells. And it also allows us to have reabsorption of nutrients in the kidneys. So if you take a look at this, they say it looks cube shaped. If you take a look, it's very hard to see a cube here, right? Instead, what I want you to memorize here is that when we're looking at simple cuboidal cell, take a look at how big the nucleus are. The nucleus, here's one cell with one nucleus. Here's another cell with another nucleus. Here's another cell with another nucleus. See how that goes? Right? Each nucleus means another cell. So these are there's like 15 to 20 different cells on this side. Here's another 15 to 20 different cells on the other side. How can you tell simple cuboidal? It's just how large the nucleus is. Nice, huge, round nucleus. Looks like a beach ball, right? It almost covers up all of the cell. See how much of that cell is covered by the nucleus? That's how you can tell simple cuboidal epithelium. You're looking for nice, big, round nuclei that almost makes up 3/4 of the cell. It takes up 3/4 of the cell. The next one, simple columnar epithelium. When you look at simple columnar epithelium, no location, bronchioles of the lungs, they're found in the uterine tubes, stomach, and intestine. When they're found in the bronchioles and the uterine tubes, they usually have cilia attached to it. When they're found in the intestines, the stomach and intestines, right, they have microvilli attached to it. Why? Different functions, right? Microvilli going to be there for increased absorption. Of course, where do we see it? You'll see it in the cells of the intestines. Cilia is going to move something. It can move phlegm. It can move sputum in the bronchioles of the lung. Or it can move an ovulated oocyte when we find it in the fallopian tubes or uterine tubes. So depending on where we see it and what kind of structure is associated with it, we'll have the different functions. If we see ciliated simple columnar cells in the bronchioles, it's there to move some kind of mucus out. If we see ciliated simple columnar in the uterine tubes, it's there to move the oocyte into the uterus. If we see it in the stomach and intestines, it's there for secretion and absorption, and usually it has microvilli attached to it. Now, what does it look like on a microscope? Because eventually, we'll talk about more lecture material next week because we have the lecture exam next week. But, in 2 weeks, we're going to really look at the histology and look at the bones, right? And discuss it. And it'll be more lab stuff as we get ramped up for the lab practical in a few weeks. So if you take a look at simple columnar cells, take a look at how long those cells are. Also, take a look at the nucleus here. The nucleus, they don't look flat like we see in simple squamous. The nuclei, here's one nucleus, here's another, right? So the nuclei here look very oval shaped, like a football. In simple cuboidal, the nucleus looks like a beach ball, big round nucleus. Here, it's oval shaped, right? And it's stacked right next to each other. You can draw a line between one nucleus to the next. They're right at the same area from one cell to the next. If you see this pink spot right here, this pink area looks like little brushes, right? It has lots of cilia, right? And that's what another way to tell. First way to tell, look at the nuclei. If you see a lot of nucleus that are oval shaped, immediately, I want you to think simple columnar cell, right? It's a sign that the cell is longer than it is wide. That's one way to tell. We'll go through how you can differentiate in a couple weeks, right? The last one we'll cover, and then we're done. Stratified squamous epithelium. Stratified squamous epithelium, there's two types. There are ones that are keratinized, making it tougher, more dense. Think of the skin at the palms or at the soles of our feet that gets real rough, right? If you walk on your feet a lot, or you're barefoot a lot, you're gonna have real rough skin on the bottom of your feet, the sole of your feet. That roughness is due to increased amount of keratin. We'll talk more about skin also in chapter 5 next week, right? It can be keratinized or moist. The moist ones does not have keratin, right? What we're going to see with stratified squamous cells is this. Its primary job is to protect. It protects against cuts and abrasions. It protects against caustic chemicals, right? It protects against water loss and even infection. It protects. How does it protect? It protects because there's so many layers that when you get a cut, sometimes you don't even cut through all the layers, right? Think of what happens when you get a scrape and you get, right? Let's say you're diving for something, playing basketball or football, and then you land and you scrape up your elbow. It might not bleed, but it hurts like crazy. Why? Because you have multiple layers here. You might scrape off some of the layers, but you don't damage the blood vessels underneath. That's why you don't bleed, right? The moist form, there's no keratin. That's what we see here. We see it in the mouth, the throat, the larynx. So at the start of the GI tract, at the end of the GI tract, and in the female reproductive system, right? With the moist type of stratified squamous cell, you still have protection, but it's moist enough that you can have ease when you're swallowing and drinking food. So that's why we see it in the mouth, throat, and the larynx right here. Protection, yet supple enough to allow for swallowing. That's why the moist is so important. Now, if you had keratinized form and it's in your mouth and throat, it'd be similar to swallowing water when you have a really bad sore throat. We've all had that real bad sore throat. You went to sleep, and then when you wake up, it hurts like crazy. It hurts so much that you know you have to drink water, but you know it's going to hurt as well just drinking water. That's what it'd be like if we had the keratinized form, right? We don't want that. We want instead the moist stratified squamous epithelial cells. So then there's greater ease when you're swallowing. It allows food. It allows boluses of drink to go slide down the esophagus with greater ease. At the same time, it's still stratified, so you have protection. Best of both worlds. All right, I'll stop right here because I think we've been going for 2 hours and 15 minutes. So